यूनिट 8 बायोलॉजी इन ह्यूमन वेलफेयर ह्यूमन हेल्थ एंड डिसीज 8 नंबर का चैप्टर है हमारा माइक्रोब्स इन ह्यूमन वेलफेयर. Biology is the youngest of the formalized disciplines of natural science. Progress in physics and chemistry proceeds much faster than in biology. Applications of physics and chemistry are in actually life also have a higher visibility than those of biology. However, 20th century and certainly 21st century has demonstrated the utility of biological knowledge in the for furthering human welfare, be it in health sector or agriculture. The discovery of antibiotics and synthetic plant derived drugs and anesthetics have changed the medical practice on one hand, and human health on the other hand. Life expectancy of human beings have dramatically changed over the years. Agriculture practices, food processing, and diagnostics have brought cultural changes in human communities. These are briefly described in the following the three chapters of this unit. Born in August 1925 in Kumba Konam in Tamil Nadu, Monkambu Sambhavan Swaminathan did his graduation and post graduation in botany from Madras University. He worked in different capacities in large number of institutions in India and abroad, and developed his expertise in genetics and plant breeding. The school of cytogenetics and radiation research established at the Indian Agricultural Research Institute (IRI) enabled Swaminathan and his team to develop short duration high yielding variety of rice including centered basmati. He is also known for the development of the concept of crop cafeteria, crop scheduling, and genetically improving the yield and quality. Swaminathan initiated a collaboration with Norman Borlaug. His culminated in the Green Revolution through introduction of Mexican varieties of wheat in India. This was highly recognized and appreciated. He is also the initiator of lab to land, food security, and several other environmental programs. He has been honored with Padma Bhushan and several other prestigious awards, medals, and fellowships by institutions of excellence. 7 number का हमारा चैप्टर ह्यूमन हेल्थ एंड डिसीजेस. 7.1 कॉमन डिसीजेस इन ह्यूमंस. 7.2 इम्यूनिटी. 7.3 एड्स. 7.4 कैंसर. 7.5 ड्रग्स एंड अल्कोहल अब्यूज. हेल्थ for a long time was considered as a state of body and mind where there was a balance of certain humorous. Now this is what early Greeks like Hippocrates as well as Indian Ayurveda system of medicine asserted. It was through thought that persons with black bile belonged to hot personality and would have fevers. This idea was arrived at by pure reflective thought. The discovery of blood circulation by William Harvey using experimental method and the demonstration of normal body temperature in persons with black bile using thermometer disapproved the good humor hypothesis of health. In latter years, biology stated that mind influences through neural system and endocrine system, our immune system, and that our immune system maintain our health. His mind and mental state can affect our health. Of course health is affected by first genetic disorders, deficiencies with which a child is born, and deficiencies defects which the child inherits from parents from birth. Second, infections, and third, lifestyle including food and water we take, rest and exercise we give to our bodies, habits that we have or lack, etc. The term health is very frequently used by everybody. Do we define it? Health does not simply mean the absence of disease or physical fitness. It could be defined as state of complete physical, mental, and social well-being. When people are healthy, they are more efficient at work. This increases productivity and brings economic prosperity. Health also increases longevity of people and reduces infant and maternal mortality. Balanced diet, personal hygiene, and regular exercise are very important to maintain good health. Yoga has been practiced since time immemorial to achieve physical and mental health. Awareness about diseases and their effect on different body functions, vaccination, immunization against infectious diseases, proper disposal of waste, control of vectors, and maintenance of hygiene in food and water resources are necessary for achieving good health. When the functioning of one or more organs or systems of the body is adversely affected, characterized by appearance of various signs and symptoms, we say that we are not healthy, that is we have a disease. Diseases can be broadly grouped into infectious and non-infectious. Diseases which are easily transmitted from one person to another are called infectious diseases. Infectious diseases are very common and every one of us suffers from these at some time or other. Some of the infectious diseases like AIDS are fatal. Among non-infectious diseases, cancer is the major cause of death. Drugs and alcohol abuse also affect our health adversely. 7.1 common diseases in humans, a wide range of organisms belonging to bacteria, viruses, fungi, protozoans, health helminthes, etc., could cause diseases in man. Such disease causing organisms are called pathogens. Most parasites are therefore pathogens as they cause harm to the most host by living in or on them. The pathogens can enter our body by various means, multiply, and they interfere with normal vital activities, resulting in morphological and functional damage. Pathogens have to adapt to life within the environment of the host. For example, the pathogens that enter a gut must know a way of surviving in the stomach at low pH and resting the various digestive enzymes. A few representative members from different groups of pathogenic organisms are discussed here along with the diseases caused by them. Preventive and control measures against these diseases in general are also briefly described. Salmonella typhi. Salmonella typhi is a pathogenic bacterium which causes typhoid fever in human beings. These pathogens generally enter the small insectine through food and water contaminated with them and migrate to other. Balanced diet, personal hygiene, and regular exercise are very important to maintain good health. Yoga has been practiced since time immemorial to achieve physical and mental health. Awareness about diseases and their effect on their on different bodily functions, vaccination, immunization against infectious diseases, proper disposal of waste, control of vectors, and maintenance of hygiene in food and water resources are necessary for achieving good health. When the functioning of one or more organs or systems of the body is adversely affected, characterized by appearance of various signs and symptoms, we say that we are not healthy, that is we have a disease. Diseases can be broadly grouped into infectious and non-infectious diseases. uh, Diseases which are easily transmitted from one person to another are called infectious diseases. Infectious diseases are very common and every one of us suffers from this at some time or other. Some of the infectious diseases like AIDS are fatal. Among non-infectious diseases, cancer is the major cause of death. Drugs and alcohol abuse also affect our health adversely. 7.1 common diseases in humans. A wide range of organisms belonging to bacteria, viruses, fungi, protozoans helminths, etc., could cause diseases in man. Such diseases causing organism are called pathogens. Most parasites are therefore pathogens as they cause harm to the host by living in or on them. The pathogens can enter our body now by various means, multiply, and they interfere with normal vital activities, resulting in morphological and functional damage. Pathogens have to adapt to life within the environment of the host. For example, the pathogens that enter the gut must know way of surviving when the stomach of at low pH and resisting the various digestive surviving various digesting enzymes. A few representative members from different groups of the pathogenic organisms are discussed here, along with the diseases caused by them. Preventive and control measures against these diseases in general are also briefly described. Salmonella typhi is a pathogenic bacterium which causes typhoid fever in human beings. These pathogens generally enter the small intestine through food and water contaminated with them and migrate to other organs through blood. Sustained of high fever, 39 degrees Celsius and 40 to 40 degrees Celsius, weakness, stomach pain, constipation, headache, and loss of appetite are some of the common symptoms of this disease. Intestinal perforation and death may occur in several causes. Typhoid fever could be confirmed by vital test. A classic case in medicine that of Mary Mellon, nicknamed of Typhoid Mary, is worth mentioning, where she was to cook by profession and was typhoid carrier who continued to spread typhoid for several years through the food she prepared. Bacteria of like Streptococcus pneumonia and Haemophilus influenza are responsible for the disease pneumonia in humans, which infects the valvular air filled sacs of the lungs. As a result of the infection, the alveoli get filled with fluid, leading to several problems in respiration. The symptoms of pneumonia include fever, chills, cough, and headache. In several cases, the lips and fingernails may turn gray to bluish in color. The healthy person acquires the infection by inhaling the droplets, aerosols released by the infected person, or even by sharing the glasses or utensils with an infected person. Dysentery, plague, diphtheria, etc., are some of the other bacterial diseases in man. Many viruses also cause diseases in human beings. Rhinoviruses represent one such group of viruses which cause one of the most infectious human ailments, and the common cold. They infect the nose and respiratory passage but not the lungs. The common cold is characterized by tired nasal congestion and discharge, sore through hoarseness, cough, headache, tiredness, etc., which usually last for 7 days. Droplets resulting from cough or sneezes of an infected person are either inhaled directly or transmitted through contaminated objects such as pens, books, cups, door knobs, computer keyboard, or mouse, etc., and cause infection in a healthy person. Some of the human diseases are caused by protozoans too. You might have heard about malaria. The disease has been fighting since many species of Plasmodium, vivax, and P. malaria and P. falciparum are responsible for different types of malaria. Of this, malignant malaria caused by Plasmodium falciparum is the most serious one and can even be fatal. Let us take a glance at the life cycle of Plasmodium. Figure 7.1. Plasmodium enters the human body as sporozoites, the infectious form. infectious form and through the bite of infected female and Anopheles mosquito. The parasites initially multiply within the liver cells and then attack the red blood cells, the RBCs, resulting in their rupture. The rupture of RBCs. Tundi. The rupture of RBCs is associated with release of toxic substance, hemozion, which is responsible for the chill. Mosquito bites an infected person. These parasites enter the mosquito's body and undergo further development. The parasites multiply within them to form sporozoites that are stored in their salivary glands. When these mosquitoes bite a human, the sporozoites are introduced into his or her body, thereby initiating the events mentioned above. It is interesting to note that the malarial parasite requires two hosts, human and mosquitoes, to complete its life cycle. Figure 7.1. The female Anopheles mosquito is the vector transmitting agent to Entamoeba histolytica is a protozoan parasite in the large intestine of human, which causes amoebiasis, amoebic dysentery. Symptoms of this disease causes includes constipation, abdominal pain, and cramps, stools, with excess mucus and blood clots. House flies act as mechanical carriers and serve to transmit the parasite from the surfaces of an infected person to food and food products, thereby contaminating them. Drinking water and food contaminated by the fecal matter are the main source of infection. Ascaris, the common round half worm and Vaucheria, and the filarial worm are some of the helminthes which are known to be pathogenic to man. Ascaris, an intestinal parasite, causes ascariasis. Symptoms of these diseases include internal bleeding, muscular pain, fever, anemia, and blockage of the intestinal passage. The eggs of the parasite are excreted along with the feces of infected persons, which contaminate soil, water, plants, and etc. A healthy person acquires this infection through contaminated water, vegetables, fruits, etc. Vaucheria, W. Bancrofti, and W. Malay. The filarial worms cause slowly developing chronic inflammation of the organs in which they are live for many years, usually lymphatic vessels of the lower limbs, and disease is called elephantiasis and filariasis. Figure 7.2. The genital organs are also often affected, resulting in gross deformities. The pathogens are transmitted to a healthy person through the bite by the female mosquito vectors. Many fungi belonging to the genera Microsporum, Trichophyton, and Epidermophyton are responsible for ringworms, which is one of the most common infectious diseases in man. Appearance of dry, scaly lesions on various parts of the body, such as skin, nails, and scalp. Figure 7.3 are the main symptoms of the disease. These lesions are accompanied by intense itching. Heat and moisture help these fungi to grow, which makes them thrive in skin folds, such as those in the groin or between the toes. Ringworms are generally acquired from soil or by using towels, clothes, or even the comb of infected individuals. Maintenance of personal and public hygiene is very important for prevention and control of many infectious diseases. Measures for personal hygiene include keeping the body clean, consumption of clean drinking water, food, vegetables, fruits, etc. Public hygiene includes proper disposal of waste and excreta, periodic cleaning and disinfection of waste reservoirs, pools, cess of pools and tanks, and observing standard practices of hygiene in public catering. These measures are particularly essential where the infectious agents are transmitted through food and water such as typhoid, amoebiasis, and ascariasis. In cases of airborne diseases such as pneumonia and common cold, in addition to the above four measures, close contact with the infected persons or their belongings should be avoided for diseases such as malaria and filariasis that are transmitted through insect vectors. The most important measure is to control or eliminate the vectors and their breeding places. This can be achieved by avoiding stagnation of water in and around the residential areas, regular cleaning of household coolers, use of mosquito nets, introducing fishes like gambas in ponds that feed on mosquito larvae, spraying of insecticides in dishes, drainage areas, and swamps, etc. In addition, doors and windows should be provided with wire mesh to prevent the entry of mosquitoes. Such such precautions now have become more important, especially in the light of recent widest widespread incidences of the vector-borne Aedes mosquitoes diseases like dong, dra- dengue, and chikungunya in many parts of India. The advancements made in biological science have armed us to effectively deal with many infectious diseases, and the use of vaccines and immunization programs have enabled us to completely eradicate the deadly disease like smallpox, a large number of other infectious diseases like for polio, diphtheria, pneumonia, and tetanus have been controlled to a large extent by the use of vaccines, biotechnology, about which you will read more in chapter 10, is at the verge of making available never and suffer vaccines. Discovery of antibiotics and various other drugs has also enabled us to effectively create infectious diseases. 7.2 immunity. Every day we are exposed to large number of infectious agents. However, only a few of them exposures result in disease. Why this is due to a fact that the body is able to defend itself from most of these foreign agents. This overall ability of the host to fight the disease causing organisms conferred by the immune system is called immunity. Immunity is of two types: innate immunity and acquired immunity. Innate immunity is non-specific type of defense and that is present at the time of birth. That is, this is accomplished by providing different types of barriers to the entry of the foreign agents into our body. Innate immunity consists of four types of barriers, and these are physical barriers. Skin on our body is the main barrier which prevents entry of the microorganisms. Mucus coating of the epithelium lining our respiratory, gastrointestinal, and urogenital tracts also help in trapping microbes entering our body. Physiological barriers: acid in the stomach, saliva in the mouth, and tears from eyes all prevent microbial growth. Cellular barriers: certain types of leukocytes of WBC of our body, like polymorphonuclear leukocytes, PMNL, neutrophils, and monocytes, the unnatural bacterial type of lymphocytes in the blood, as well as macrophages in tissues, can phagocytose and destroy microbes. Cytokine barriers: virus infected cells secrete proteins called interferons, which protect non-infected cells from further viral infection. 7.2.2 acquired immunity. Acquired immunity, on the other hand, is pathogen specific. It is characterized by memory, This means when our body encounters a pathogen for the first time, it produces a response called primary response, which is of low intensity. Subsequent encounter with that same pathogen elicits a highly intensified secondary or anamnestic response. This is ascribed to the fact that our body appears to have memory of the first encounter. The primary and secondary immune responses are carried out with the help of two special types of lymphocytes present in our blood, that is B lymphocytes and T lymphocytes. The B lymphocytes produce an army of proteins. It responds to pathogens into our blood to fight with them. These proteins are called antibodies. The T cells themselves do not secrete antibodies but help B cells to produce them. Each antibody molecule has four peptide chains, two small called light chains and two longer called heavy chains. Hence, an antibody is represented by H2L2. The different types of antibodies are produced in our body: IgA, IgO, IgE, IgG are some of them. A cartoon of an antibody is given in figure 7.4. Because these antibodies are found in the blood, the response is also called a humoral immune response. This is one of the two types of our acquired immune response: antibody mediated. The second type is called cell mediated immune response or cell mediated immune CMI. The T lymphocytes mediate CMI very often when some human organs like heart, eye, liver, kidney fail to function satisfactorily. Transplantation is the only remedy to enable the patient to live a normal life. Then a search begin to find a suitable donor. Why is it that the organs cannot be taken from just anybody? Now, what is it that the doctors check? Grafts from just any source, an animal, another primate, or any human beings cannot be made, since the grafts would be rejected sooner or later. Tissues matching, blood group matching are essential before undertaking any graft or transplant, and even after this, the patient has to take the immune suppressants all his or her life. The body is able to differentiate self and non-self, and the cell mediated immune response is responsible for the graft rejection. 7.2.3. Active and passive immunity. When a host is exposed to antigens, which may be in the form of living or dead microbes or other proteins, antibodies are produced in the host body. This type of immunity is called active immunity. Active immunity is slow and takes time to give its full effective response. Injecting the microbes deliberately during immunization of infectious organisms, gaining access into body during natural infection induce active immunity. When ready-made antibodies are directly given to protect the body against foreign agents, it is called passive immunity. Do you know why a mother's milk is considered very essential for the newborn of infant? The yellowish fluid colostrum secreted by mother during the initial days of lactation has abundant antibodies IgA. Now, to protect the infant, the fetus also receives some antibodies from their mother through the placenta during pregnancy. These are some examples of passive immunity. 7.2.4. Vaccination and immunization. The principle of immunization or vaccination is based on the property of memory of the immune system. In vaccination, the preparation of antigenic proteins of pathogen are inactivated, weakened pathogen vaccine are introduced into the body. The antibodies produced in the body against these antigens would neutralize the pathogenic agents during actual infection. The vaccines also generate memory B and T cells that recognize the pathogen quickly on subsequent exposure, and other when overwhelm the invaders with the massive production of antibodies. If a person is infected with some deadly microbes to which quick immune response is required. As in tetanus, we need to directly inject the performed antibodies or antitoxin preparation containing antibodies to the toxin. Even in cases of snake bites, the injection which is given to the patients contain performed antibodies against the snake venom. No, this type of immunization is called passive immunization. Recombinant DNA technology has allowed the production of antigenic polypeptides of pathogen and bacteria or yeast. Vaccines produced using this approach allow large scale of production and hence greater availability for immunization. Example, hepatitis B vaccine produced from yeast allergies. When you have gone to a new place and suddenly you started sneezing, wheezing for no explained reason, and when you went away your symptoms disappeared. Did this happen to you? Some of us are sensitive to some particles in the environment. The above mentioned reaction could be because of allergy to pollen, mites, etc., which are different in different places. The exaggerated response of the immune system to certain antigens present in the environment is called allergy. The substances to which the such immune response is produced are called allergens. The antibodies produced to this are the of IgE type. Common examples of allergens are mites in dust, pollens, dander, animal lender, etc. Symptoms of allergic reactions include sneezing, watery eyes, running nose, and difficulty in breathing. Allergy is due to release of chemicals like histamine and serotonin from the mast cells. For determining the cause of allergy, the patient is exposed or injected with very small doses of possible allergens, and the reactions studied. The use of drugs like antihistamine, adrenaline, and steroids quickly reduce the symptoms of allergy. Somehow modern day lifestyle has resulted in lowering of immunity and some more sensitivity to allergens. More and more children in metro cities of India suffer from allergens and asthma due to sensitivity to the environment. This could be causes because of the protective environment provided early in life. Autoimmunity, memory-based acquired immunity evolved in higher vertebrates based on the ability to differentiate foreign organisms, example pathogens from self cells. While we still do not understand the basis of this, two corollaries of this ability have to be understood. One, higher vertebrates can distinguish foreign molecules as well as foreign organisms. Most of the experimental immunology deals with this aspect too, sometimes due to genetic and other unknown reasons. The body attacks self cells. This results in damage of the other of the to the body and is called autoimmune disease. Rheumatoid arthritis. Rheumatoid arthritis, which affects many people in our society, is an autoimmune disease. Immune system in the body. The human immune system consists of lymphoid organs, tissue cells, and soluble molecules like antibodies. As you have read, immune system is unique in the sense that it recognizes foreign antigens, responds to this and remembers them. Uh, the immune system also plays an important role in allergic reactions, autoimmune diseases, and for organ transplantation, lymphoid organs. These are the organs where origin and maturation and proliferation of lymphocytes occur. The primary lymphoid organs are a bone marrow and thymus, where immature lymphocytes differentiate into antigen sensitive lymphocytes. After maturation, the lymphocytes migrate to secondary lymphoid organs like spleen, lymph nodes, tonsils, pairs, patches of small intestine, and appendix. The secondary lymphoid organs provide the sites for interactions of lymphocytes with the antigen, which, when proliferation to become effector cells. And the location of various lymphoid organs in the human body is known is shown in figure 7.5. The bone marrow is the main lymphoid organ where all blood cells, including lymphocytes, are produced. The thymus is a lobed organ located near the heart and beneath the breastbone. The thymus is quite large at the time of birth, but keeps reducing in size with the age, and by the time puberty is attained, it reduces to a very small size. Both bone marrow and the thymus provide microenvironments now for the development of maturation of T lymphocytes. The spleen is a large bean-shaped organ. It mainly contains lymphocytes and phagocytes. It acts as a filter of the blood by trapping blood-borne microorganisms. Spleen also has a large reservoir of erythrocytes. The lymph nodes are small solid structures located at different points along the lymphatic system. Lymph nodes serve to trap the microorganisms or other antigens which have happen to get into the lymph and tissue fluid. Antigens trapped in the lymph nodes and are responsible for the activation of lymphocytes present there and cause the immune response. There is lymphoid tissue also located with the lining of the major the tracts, respiratory, digestive, and urogenital tracts, so called mucosa associated lymphoid tissue MALT. It constitutes about 50% of the lymphoid tissue in human body. AIDS, the world the AIDS stands for say acquired immunodeficiency syndrome. So this means the deficiency of immune system acquired during the lifetime of an individual, indicating that it is not a congenital disease. Syndrome means a group of symptoms. AIDS was first reported in 1981, and in the last 25 years or so, it has spread all over the world, killing more than 25 million persons. AIDS is caused by the human immunodeficiency virus HIV, the member of a group of viruses called retrovirus, which have an envelope enclosing the RNA genome. Figure 7.6. Transmission of HIV infection generally occurs by sexual contact with the infected person. B, by transfusion of contaminated blood and blood products. C, by occurring by sharing the infected needles, as in case of intravenous drug abusers. And D, from infected mother to her child through placenta. So people who are at high risk of getting this infection includes individuals who have multiple sexual partners, drug addicts who take drugs intravenously, individuals who require repeated blood transfusions, and the children born to the HIV infected mother. Do you know when do people need repeated blood transfusion? Find out and make a list of such conditions. It is important to note that HIV/AIDS is not spread by mere touch or physical contact. It spreads only through body fluids. It is hence imperative for the physical and psychological well-being that the HIV/AIDS infected persons are on the infection and appearance of AIDS symptoms. This period may vary from a few months or to many years, usually 5 to 10 years. After getting into the body of the person, the viruses enters into macrophages where RNA genome of the virus replicates to form viral DNA with the help of the enzyme reverse transcriptase. The viral DNA gets incorporated into host cells DNA and directs the infected cells to produce virus particles. Figure 7.6. The macrophages continue to produce virus and in this way acts like a HIV and produce progeny viruses. The progeny viruses released in the blood attacks or other helper T lymphocytes. This is repeated leading to a progressive disease decrease in a number of helper T lymphocytes in the body of the infected person. During this period, the person suffers from bouts of fever, diarrhea, and weight loss. Due to decrease in number of helper T lymphocytes, the person starts suffering from infections that could be have been otherwise overcome, such as parasites like taxoplasma. The patient becomes so immunodeficient that used a diagnostic test for AIDS in enzyme-linked immunosorbent assay, ELISA. Treatment of AIDS with antiretroviral drugs is only partially effective. They can only prolong the life of the patient but cannot prevent death, which is inevitable. Prevention of AIDS. As AIDS has no cure, prevention is the best option. Moreover, HIV infection more often spreads due to conscious behavior patterns and is not something that happens inadvertently, like pneumonia or typhoid. Of course, infection in blood transfusion patterns patients newborns may be ignored, and it has been rightly said, don't die of ignorance. In our country, the National AIDS Control Organization NSCO and other non-government organization NGOs are they are doing a lot of educate people about the AIDS. Who WHO has started a number of programs to prevent the spreading of HIV infection, making blood from blood banks safe from HIV, ensuring the use of only disposable needles and syringes in public and private hospitals and clinics, free distribution of condoms, controlling drug abuse, advocating safe sex, and promoting regular checkups for HIV in susceptible populations are some such steps taken up. The infection with HIV and having AIDS is a something that should not be hidden, since then the infection may spread to many more people. HIV/AIDS infected people need help and sympathy instead of being stunned by society. Unless society recognizes it has a problem to be dealt with in a collective manner, the chances of wider spread of the disease increase manifold. It is a malady that can only be tackled by the society and medical fraternity and acting together to prevent the spread of disease. Cancer is one of the most most dreaded diseases of human beings and is a major cause of death all over the global. More than half a million Indians suffer from cancer, and a large number of them die from it annually. The mechanisms that underlie development of cancer or oncogenic transformation of cells, its treatment and control have been some of the most intense areas of research in biology and medicine. In our body, cell growth and differentiation is highly controlled and regulated. In cancer cells, there is breakdown of these regulatory mechanisms. Normal cells show a property called contact inhibition, by virtue of which contact with other cells inhibits their uncontrolled growth. Cancer cells appear to have lost this property. As a result of this, cancerous cells just continue to divide and giving rise to masses of cells called tumors. Tumors are of two types, being benign and malignant. Benign tumors normally remain confined to their original location and do not spread to other parts of the body and cause little damage. The malignant tumors are, on the other hand, are a mass of proliferating cells called neoplastic or tumor cells. These cells grow very rapidly, invading and damaging the surrounding normal tissues. As these cells actively divide and grow, they starve the normal cells by competing for vital nutrients. Cells sloughed from such tumors reach in distant sites through blood, and wherever they are get lodged in the body, they start a new tumor. Here they are. Now this property called metastasis now is the most feared property of malignant tumors. Causes of cancer: transformation of normal cells into cancerous neoplastic cells may be induced by physical, chemical, or biological agents. These agents are called carcinogens. Ionizing radiations like X-rays and gamma rays, and non-ionizing radiations like UV cause DNA damage, leading to neoplastic transformation. The chemical carcinogens present in tobacco smoke have been identified as a major cause of lung cancer. Cancer causing viruses called oncogenic viruses have genes called viral oncogenes. Furthermore, several genes called cellular oncogenes C1 and proto-oncogenes have been identified in normal cells, which, when activated under certain conditions, could lead to oncogenic transformation of the cells. Cancer detection and diagnosis: early detection of cancer is essential as it allows the diseases to be treated successfully. In many cases, cancer detection is based on biopsy and histopathological studies of the tissue and blood and bone marrow tests for increased cell counts. In the case of leukemias, in biopsy, a piece of the suspected tissue cut into thin sections, is stained and examined under microscope. Histopathological studies by pathologists say techniques like radiography, use of X-ray, CT, computed tomography, and MRI, magnetic resource imaging are very useful to detect cancers of the internal organs. Computed tomography uses X-rays to generate a three-dimensional image of the internals of an object. MRI uses strong magnetic fields and non-ionizing radiations to accurately detect pathological and physiological changes in the living tissue. Antibodies against cancer-specific antigens are also used for detection of certain cancers. Techniques of molecular biology can be applied to detect genes in individuals with inherited susceptibility to certain cancers. Identification of such genes which predispose and identical to a certain cancers may be very helpful in prevention of cancers. Such individuals may be advised to avoid exposure to particular carcinogens to which they are susceptible. Example: tobacco smoke in case of lung cancer. Treatment of cancer: the common approaches for treatment of cancer are surgeries, radiation therapy, and immunotherapy. In radiotherapy, tumor cells are irradiated lethally taking proper care of normal tissues surrounding the tumor mass. Several chemotherapeutic drugs are used to kill cancerous cells. Some of these are specific for particular tumors. Majority of drugs have side effects like hair loss, anemia, etc. Most cancers are treated by a combination of surgery, radiotherapy, and chemotherapy. The immune system, therefore, the patients are given substances called biological response modifiers such as interferon, which activates their immune system and helps in destroying the tumor. 7.5 drugs and alcohol abuse. Skeletal structure of cannabinoid molecule. 7.10 leaves of cannabis sativa. 7.11 flowering branch of datura. Adolescents and drugs, alcohol abuse, addiction and dependence. 7.5.3 effect of drugs and alcohol abuse. 7.5.4 prevention and control. Prevention and control, avoid undue peer pressure, education and counseling, seeking help from parents and peers, looking for danger signs, seeking professional and medical help. Now, human health and diseases summary. Human health and diseases 7 नंबर का हमारा चैप्टर है ह्यूमन हेल्थ एंड डिसीजेस, कॉमन डिसीजेस इन ह्यूमंस. 7.1 कॉमन डिसीजेस इन ह्यूमंस. 7.2 immunity, acquired immunity, active and passive immunity, vaccination and immunization, allergies, autoimmunity, immune system in the body. 7.3 AIDS. 7.4 cancer. 7.5 drugs and alcohol abuse. 7.5.1 adolescents and drug alcohol abuse. 7.5.2 addiction and dependence. 7.5.3 the effects of drug and alcohol abuse. 7.5.4 prevention and control. Then summary. Please like, share and subscribe. Thank you so much for watching.